Okay, so hello everybody, and welcome again to the second part of the meeting. Unfortunately, Teresa had a problem, has to travel, so she asked me to step in. Um, so the idea is more or less going with the same dynamics than in the previous session, and just in trying to keep the time, I will skip very long introductions and just give the floor to Guilherme, not without thanking him for the hard work of putting together all these meetings already, because if you ignore that he has done almost everything by himself, so it's a lot of work. Thank you very much, Guilherme. I can grant you an extra minute for that. So whenever you want.
Hello, everybody. I'm going to talk about modeling the reactivity of metalloproteins. I am Guilherme Arantes from the University of São Paulo. It's based in this concrete jungle, one of the largest cities in the Southern Hemisphere. I run this computational biophysics and biochemistry lab. So metalloproteins are very important. Most of the metals, they have structural and stabilization roles, but particularly transition metals perform unique chemistry, and they're very important for reactivity. For instance, iron sulfur clusters are the most abundant metal cofactors. They are ubiquitous in cellular redox processes. Today, I'll be talking about two metalloproteins, hubredoxin, which is a simple iron sulfur protein with one iron center coordinated to four sulfur atoms of the side chains of four cysteines with a tetrahedral coordination. Its role is to transfer electrons. The other protein I'll be talking about is metallothionein. It's composed of two domains. I'll be talking only about alpha domain, which contains a cluster of four metal centers bound by 11 cysteine residues, forming this cluster here, showing in the right, with two fused six-membered rings. It's a very unique polynuclear metal cluster. So how do we study these proteins? We use molecular simulation, of course, with hybrid quantum chemical molecular mechanical potentials. We do a structural and energetic partitioning of our system. For instance, if we have a pictorial view of the protein, let's say the hubredoxin, with the active side, the metal center is treated with quantum mechanics, with quantum chemistry that's necessary to model bond breaking, bond forming, for instance. All the remaining of the protein and the solvent, they are treated with more approximate molecular mechanics force fields. So the Hamiltonian is written as a combination of the quantum chemical part, the molecular mechanical part, and this coupling between the two regions. The main ingredient here for the hybrid potential is this coupling that allows partial charges in the molecular mechanical region to interact to polarize the electronic density of the quantum chemical region. So we start showing the results for metallothionein. This structure was determined by an MR, and it's interesting that the apo metallothionein without any metal bond is an intrinsically disordered protein. So the protein is very flexible. This protein is involved in metal homeostasis. It will bind several different metals, mostly zinc, to transport. We were interested in understanding the stability of this metal cluster. So using our hybrid potential with the metals and the cysteine side chains modeled by DFT, we calculated the energy to reconfigure the cluster. The central structure here is the structure found by NMR, and if a bond is formed between the cysteine and zinc's number 6, we'll see this structure being formed. It's shown here in orange, and it has a quite low barrier, showing that this process can easily happen. If we run molecular dynamics simulations, we will see spontaneously bond breaking and bond forming with a cluster reconfiguration in the picosecond time scale. So those properties are important because metallothionein is a metal carrier. So it's important that it binds to metals fast and release the metals when necessary. The second metalloprotein we studied, ubiredoxin, we would like to understand, again, the reactivity, the stability of iron-sulfur bond. Two reaction mechanisms can be observed. A homolytic cleavage, where the electron pair of the bond broken will be split, and a heterolytic mechanism, where the electron pair of the broken bond will stay in the thiolate. Also, in the ground state, the spin state is a sextet. We would like to know, will it always stay in the sextet state when it reacts, or can it cross to another spin state, to lower spin states? So for rubiredoxin, folded, the iron center is kind of buried in the protein, so water cannot access the iron center. We studied then the bare dissociation, breaking the iron-sulfur bond without any substitution into iron. We use our quantum chemical, molecular mechanical hybrid potential, again, with the metal and the cysteine side chains described by DFT, but this time iron has an open shell. Because no solvent can access the iron center and the proteins constrain the reaction, we find a high barrier here, quite high, higher than 150 kilojoules per mole. But interestingly, we find that the product is homolytic with the electron pair split between iron and sulfur. And although the reactant state is in a sextet, the product is in a quartet. So we find spin crossing in the reaction cores. But what if we can unfold the protein and allow water to access the iron center? How is the reaction like? We tested several mechanism proposals for two consecutive iron-sulfur ruptures by hydrolysis in several protonation states. In neutral medium, so without any extra protonation of the living groups, we find quite high barriers as well, higher than 100 kilojoules per mole for the two consecutive reactions that are necessary to fully unfold the protein. With protonation, with single protonation, the reaction for this step is slower, but the second step is about 80 kilojoules per mole. It should be noted that all those reactions for water substitution will stay in the sextet state because the quartet is always higher in energy. And the mechanism is heterolytic. The electron pair will stay with the thiolate. We can compare those results to experiment. The activation energy obtained from experiment is about 80 kilojoules per mole, and the protein will refolding spontaneously. So the barrier for the reaction from the right to the left has to be low, as we find here. In a highly acidic media, if we have a protonation of all the cysteines, we'll see barriers lower than 20 kilojoules per mole. That's related to why the cluster, the iron-sulfur cluster, is buried inside the protein, because this will increase its stability against hydrolysis, against acid catalyzed hydrolysis. For all these reactions, we find that an associative mechanism is preferred. Here I'm showing the transition state for the first water substitution, and you can see that the iron here in orange and the sulfur in yellow, they have already a broken bond, and the bond between the water, oxygen, and iron is being formed. Both bond breaking and bond forming will occur at the same time or simultaneously. So this is what we call an associative mechanism. It's the preferred mechanism for all protonation states we have studied. I would like to finish thanking my collaborators, Felipe and Murilo from my group here in the University of São Paulo, and Martin Field from France, and Feng Zhang from China, that are co-authors of the studies. Also would like to thank FAFEST for the money, and to announce that we have opportunities for graduate students and postdocs in our group. Thank you very much for your attention.
All right. Thank you very much, Guilherme. So we move to the next speaker, who is Mauricio Coutinho.
Hey, Sergio. I would like to thank Guilherme. I'm going to try an SBBq. I'm going to try to share my screen here. Share sound and optimize for video. I never done that. Let's see if this works. So hello, everybody. First, I would like to thank Guilherme for putting this workshop together and you for your attendance. My name is Mauricio Domingues Coutinho Neto, and I'm going to talk today about selenium and tellurium calculate compounds as warheads for protein inhibition. And I'm from UFABC, which is a federal university in the ABC region, São Paulo, Brazil. And I'm going to first talk a little bit about myself. So we are located in the southern part of São Paulo. So this is the urban mass of São Paulo. We are located around here. And UFABC is a new institution. It was created in 2006. And this is our main building in 2009. This is present UFABC, actually 2019. And we have around almost 15,000 undergrad students and almost 2,000 graduate students, several undergrad and graduate granting degrees. So as I said, today I'm going to talk about substitution reaction on calculate, which are hypervalent tellurium and tellurium and selenium compounds to be used as new warheads for protease inhibition. I'm also interested in several other research topics. So here is a list of them. So if you are interested in any of them, please drop in a line if you want to discuss or collaborate. So this research project was born from collaboration with Rodrigo Cunha. So Rodrigo Cunha is a synthetic organic chemist. So he synthesized these hypervalent compounds. This is tellurium 4 compounds and selenium 4 compounds. And these compounds, they show remarkable activities toward protease inhibition, in particular cysteine protease inhibition. And usually these compounds are synthesized with chlorine or bromine here to ensure their hypervalent state. And they are using aqueous solution. So the question that he had is concerning their actual the actual active form in solution. So what's interesting also about his compounds, these compounds, is that they inhibit cysteine protease through the formation of a covalent bond between the molecule, right, the drug, and the cysteine residue in cysteine proteases, which leads to a very efficient inhibition. So as I said, the main question that the experimentalist that Rodrigo had is if you start with these chlorinated compounds, they undergo ligand substitution in solution, in aqueous solution, especially in biological solution in which you have a multi-nucleophile environment. And he didn't know what's the actual form that would inhibit the enzyme. What binds to the enzyme is still unknown, and this is the main motivation of our study. So we went on and we basically did three main lines of investigation. First, we went on to investigate the thermodynamic stability of several of these compounds in solution, compounds that were formed by ligand exchange reaction starting from the dechlorinated compound. This is the main topic of my talk today. We also investigate their interconversion barriers, and later on we went on to do QMMM based on the results of the first part. Once we knew or we had a good guess on what is the actual stable form in solution, in aqueous solution, we went on and did the QMMM studies with these proposed species. So as I said, what we did on the thermodynamic part of the study is that we investigated the stability of this species in solution. So So this is the general form of this our diorganal compound, with R being CH3, methyl, and phenyl. Y is the chalcogen, so Y could be selenium and tellurium. And we consider deprotonated nucleophiles, which is what we would have in neutral basic pH, and also protonated nucleophiles, which would be the stable form, obviously, in an acidic pH. So this is how we consider pH in our study. So in the end, we investigated 35 unique species, which led to 155 reaction pre-entries. In particular, we also investigate the formation of three coordinated species. We, as you would see, turn out to be very important for this study. So the three coordinated with OH, O, SH, and chlorine, which were formed from the elimination of one ligand from the tetracoordinated species. So on to the results, what we found was that the chlorine, the tetracoordinated chlorine, is the least stable form in solution. It could undergo ligand substitution, sequential ligand substitution leading to the three coordinated species, in particular here, the selenoxide species. And this, now, at least based on this model reactions, this would be the stable form in solution, which would then react to the cysteine here, which is being modeled as SH minus group, right? And this would be the final stable form, which would lead final stable form in solution, which would in an enzyme analog would lead to the inhibition. For the tellurial compounds, we have basically the same. The reactions are more exergonic, especially the formation of the oxides and telluroxide and protonated telluroxide are very exergonic, and this would be also the species that would lead to the inhibition. This reaction substitutions could also be seen on this side diagram. This is the starting compound, and this, what we call mechanism 1 in neutral basic, which would be the favorite one in neutral basic pH, which is the pH in which we have the unprotonated nucleophiles, right? The negative charge, the stronger nucleophiles, would be either the formation of a tetracoordinated dihydroxide compound, which would then loses water, right? Dehydrates, form this protonated form here of the selenoxide, or the unprotonated form, which would quickly go here. And this would be the form that would lead to the inhibition. We also uncover a second mechanism, which would be favored in acidic solution, in which is the case in that we have the protonated nucleophiles, which would start by the loss of a chlorine atom here on the selenium compound, which would be slightly energonic. On the tellurium, this is exergonic, but this species can directly be attacked actually by the cysteine, leading to the inhibition, or it could also hydrolyze this water, forming the selenoxide or telluroxide, which would then lead to the inhibition. On this side scheme here, we have basically this guy leading directly either to this, or directly going to the forming the three coordinated chlorine species and the inhibition. So either this path or this direct path would lead to inhibition. But regardless of mechanism 1 or 2, what we found is that this species in an aqueous solution is very stable and would most probably be the one that would lead to the inhibition. We also did mechanism studies, right, kinetic studies, and the literature proposed 2 limiting mechanisms for ligand substitution on this system. The first one is elimination addition mechanism, which would involve the breakage of one of the ligand bonds, forming a three-coordinated species, and this guy is then attacked by the nucleophile, forming again a tetracoordinated one. This will be the equilibrium. And a second proposed mechanism is the addition and elimination, which would be the inverse. First, would have nucleophile attack, followed by elimination, right? So we computed several reaction barriers. I'm going to show a few of them. But what we found that was very interesting is that what we found was something between. But in particular, right, we found that the three-coordinated species, this was also already shown from the thermodynamic studies, the three-coordinated species is very stable, and the barrier, the conversion barrier between the three-coordinated and the tetra-coordinated one is very small here. For example, if you start from the dichloride one, followed by attack of an OH group, the barrier for the formation of the three-coordinated one is only 2.5 kilocalories per mole. If you do the same type of reaction starting from this OH-SH substitute the tetra-coordinated compound, the barrier is only 2 kilocalories per mole to form the selenoxide. We also investigated other reactions, in particular the dehydration reaction of the tetra-coordinated compound here, the organotetra-coordinated the hydroxide compound, and the barrier in this case is slightly larger at 9 kilocalories per mole, but also it forms a most stable selenoxide product. So both the thermodynamic and kinetic results, they point to the formation of this selenoxide species in solution or telluride species in solution, and this in a pH equals to 7 or neutral basic pH. This guy can then undergo membrane crossing on the back wall of lysosome, right, which is the natural environment of cysteino proteases, and then at this more acidic environment, this guy protonates and can be attacked by the cysteine, leading to the covalent bond formation and inhibition. We also did some QMMM, but these results have been published in the literature recently. If you are interested about them, you can check it out. You can drop me a line if you have any questions. And we also did QMMM studies. I would like to finish thanking Gabriela Diaz, which was my student that did most of this work, my collaborators, the financing agencies, and you for your attention. Thank you very much.
Thank you very much, Mauricio. And the next speaker is going to be Laura Coitiño from Universidad de la República in Montevideo, Uruguay. Hola, Laura. You're muted.
Now it's okay.
Yes. Perfect. Please go ahead.
Thank you, Sergio. And it's nice to be here sharing our work with colleagues and friends. And what I'm going to present here is part of the work we are conducting at my group at the Universidad de la República in Montevideo, Uruguay, and is centered in a very well-known enzyme that is prostaglandin GH synthase 2, and from here COX-2, and trying to get insight on the allosteric activation and also the inhibition by a particular set of molecules that are derived from fatty acids. And we are working on that. Let's see, I cannot, it's not changing. Seems like it's not working for passing the page. Let me try again. Okay, right now it's working. Sorry.
It's not
okay.
Perfect.
So we have here COX-2, and it is really studied in a profound way because of the interest from pharmacology, because it's the cause of the synthesis of prostaglandins that are signaling mediators in diseases such as cancer, cardiovascular diseases, inflammation, and pain. And it's starting from arachidonic acid and oxygen. And we have two subunits in the structure. It's a structural homodimer and a functional heterodimer, and this is one of the interesting aspects of the enzyme. And concerning the nitroarachidonic acid, is a known inhibitor of the activity of the enzyme, one of the parts, and promotes the release of one part of the enzyme we are going to see right now. I think it's not working when I choose the pointer, because it is the same situation. Okay, we have here the same structure, and we can see here the two subunits. In blue, we have what is known as the allosteric unit, and in red, the catalytic one. And each subunit has two different activities and two active sites. I don't know if you can see the pointer here in this way, but we have here the POX site, which is a peroxidase, and here the cyclooxygenase 1, and it's repeated here the two sites. The difference is that one of the subunits is catalytic, so the reactions are happening here. In the other case, you don't have the reaction, and they have a difference in the COX site structural, because the substrate is present in the catalytic one in a productive form, and in the non-catalytic, the allosteric, is in a non-productive binder. And there is also a difference in the affinity of the heme group in the two POX sites, which is in the catalytic, there is a high affinity, in the allosteric, a low affinity. So we can see here for the red one, the reactions involved, which are connected to the POX site with the presence of the heme, and the cyclooxygenase cycle here. And the connection between the two set of reactions is a tyrosyl radical that is generated in the peroxidase cycle and is starting the reaction at the COX site. So we can see here for the catalytic subunit, there is more structural information. Actually, there is plenty of structures, but although the well-known activities and the use of this enzyme as a target for developing drugs for all the diseases mentioned, there is still a need for more knowledge concerning the basis of the mechanistic roles of the two subunits. And there is a hypothesis about the mechanism that is not proven, and more structural information is needed for that. So this work is trying to produce information that is going to contribute in this sense. So for the POX activity, we have here the structure, we have the heme group, and there are several residues that are important for having the group here. In the COX activity, you have the substrate here in the productive position, and there are also a set of residues that are important. We can see here that for that, we try to see the role of the arachidonic acid as a allosteric activator of the enzyme using the homodimer, and then the role of the nitroarachidonic acid as a inhibitor of the POX activity at the catalytic site. So what we did for that was designing an strategy with different situations for the homodimer, loading with heme groups in each subunit, symmetric in the resting state, then loading with the productive and non-productive arachidonic acid in each case, then the catalytic subunit loaded with only one heme, which is, yeah, sorry?
Y la comunicación es muy buena.
Okay, I'm going to continue. And then with the arachidonic in this system and with no heme, and then the system with the inhibitor present in the catalytic unit, and in presence and in absence of the heme group. We conducted for that molecular dynamic simulation, classical, atomistic, with these characteristics here. So concerning the results, what we got was that comparing the two subunits, we couldn't find visible changes in the 3D structures, and mostly the difference are related to side chain rotations at the active sites that are influencing the hydrogen bond networks and also the other types of interactions. And this is causing a regulation by the presence of the arachidonic acids in the active site of the COX, sorry, not in the active sites, at the COX sites in the allosteric unit, is modulating the interaction with the heme distal group, and is producing the elimination of the second heme group. And so is in a way the introduction of the substrate in a non-productive way is promoting the heme group is going out of the subunit, the allosteric one. Concerning the flexibility, you can see here that comparing the two subunits, you don't have a very important changes in flexibility. So it's mostly the same when two heme groups are present, and it's almost the same when you load the enzyme with the non-productive and productive arachidonic acid. When you go to one heme present in only one of the subunits, so what you have here is changes in very particular and important residues when you have only heme. So the asymmetry is promoted because of the absence of the second heme group. And you can see here that when you have the arachidonic acid, you have a difference in the POX site. So this is sustaining the idea of an half of sites hypothesis explaining the activity and the allosteric. When you can consider the situation having no groups here, you can see that there is no differences in this case, but both subunits introduce the arachidonic acid in a non-productive way. So these are really important information that we can use to understand better the allosteric effect of the arachidonic acid here. So I'm going to be shifting this. The mechanistic insight is the most important part, that we can collect information that departing from an asymmetric homodimer with similar units, the presence of the arachidonic acids into the COX sites in a productive orientation is compatible with the heme presence. So this is the reason we have the catalytic unit. And on the other case, when the arachidonic acid is entering in a non-productive way, we are showing that heme group is promoted to go out of the structure and creates the asymmetry. And finally, for the results for the POX inhibition, what we have here is that the nitroarachidonic acid enters to the POX site and avoids the possibility of a reaction with the hydroperoxides. And on the other case, when you don't have heme groups present, is also covering the POX site, so avoiding preventing the introduction. So we have information about the way this is working, and the most important part of the work is that we provide here structural support that is needed, and from simulations to the half of sites activity and allosteric concept, And on the other hand, we are proving that the nitroarachidonic acid enters into the POC site, covering and possibly displacing the heme group and preventing any hydroperoxide to approach to the ferric part of the heme group that is starting the cycle. And more detailed studies are under progress to gain a deeper insight on the strength of the interaction. We are conducting more detailed analysis from the MD simulations, and also going further to electronic detail with UMM calculations. So I think it's all. And we have here our faculty. I want to thank Guilherme for organizing this meeting, and all of you for your attention. Thank you.
Thank you very much, Laura.
I was in doubt you were hearing because I had some noise in the middle.
No, it was perfect.
Okay.
Thank you. So we move to the next speaker, who's going to be Nina Pastor from the Universidad Autónoma del Estado de Morelos in Mexico. Nina, are you ready? You will have to unmute yourself.
Yes.
There you are.
Yes, there. Made it.
Fantastic. We can see you, we can hear you. Please go.
Oh. Okay, so what I'm going to tell you is a very young story. It's the beginning of the PhD thesis of Marco Ramirez, and it has to do with a fascinating protein. Cre is a very precise and self-sufficient recombinase. That means that it is an enzyme dedicated to cutting and splicing DNA together. It is composed of two domains, the N-terminal domain and the catalytic C-terminal domain. This thing in yellow is a tyrosine. This belongs to the family of tyrosine site-selective recombinases that carries out a nucleophilic attack on a phosphorus here, and that's how it cuts DNA. The protein is specific for a binding site called LOXP, and it is almost perfectly palindromic. So actually, two of these proteins bind to the DNA, each at its own RBE, and forming structure here. As you can see, it bends DNA and it also unwinds it a little bit. It is so precise in its cutting and pasting abilities, which are marked here by these arrows, that it has long been used as a tool for editing genomes, both in biotechnology and in biomedicine. The problem is that some toxicity remains, so not everything is written about Cre. There are on the order of 20 structures deposited in the PDB of the interaction of Cre with DNA, so we know how it talks to DNA. You can see here this beautiful electrostatic complementarity, positive for the protein and negative for the DNA. And also all the residues that talk to DNA either directly or through water molecules. Also, as the binding site is not perfectly palindromic, the interactions of the two protomers that bind to the DNA are not identical. And that means that one of the subunits binds in such a way that the catalytic tyrosine is very close to the phosphate it's supposed to attack, while the other subunit is a little bit farther away. So this we always have one active and one inactive protomer. In order to work, this is a recombinase. That means that it takes two pieces of DNA and it exchanges bits and pieces between them. So you have one duplex here and one duplex down here, and this is called a synaptic complex. Here we're looking from the N-terminal domain site, and you can see that there's an alternation of active and inactive subunits as you go around in this square. When you look at the other side of the tetramer, what you see is that one of the subunits invades the other one in a timid attempt at domain swapping. Actually, what is inserted is an alpha helix. It's the C-terminal alpha helix of the C-terminal domain that talks to its neighbor. So there is really a lot of communication between all the subunits in this tetramer. Why would we need active and inactive protomers? That has to do with the reaction that this enzyme carries out. So we have one duplex and the other duplex. This is the green guys are the active subunits and the pink ones are inactive. So if it's active, its tyrosine is close to a phosphate. It cuts, it generates a 5' OH. Now this guy will react again with the phosphate, and now we have a new connection. The same thing happens in the other strand. Okay, so instead of being connected like this, now we have what is called a holiday junction. The next thing is a magical step called isomerization, where what used to be an active subunit now is inactive, and then you do exactly the same thing all over again, and we have managed to go from 2 strands that were separated horizontally to 2 strands that are separated vertically. It is extremely efficient. It does this really fast. It is possible to trap the intermediaries through specific mutants. So the differences between the active and the inactive conformations are really small. If you superimpose all the structures that are deposited in the PDB, you can get this cluster of structures that don't surpass 2.5 angstrom RMSD between them. So the devil is in the details. If you look at what happens when you superimpose the most deviant of these conformations, you see that the N-terminal domain suffers basically no change, and the C-terminal domain has a core that doesn't move that much, but there is a loop here. Please bear in mind that it goes from 196 to 210. I'll tell you why in a second. And also the C-terminal tail, including helix N and helix M. The catalytic tyrosine lives here in helix M, and what this changing conformation does is push it closer to the phosphorus atom that is the client for the nucleophilic attack. Now, regarding this guy, turns out that the chemistry that this enzyme does is also very well understood, and you need lysine 201, which is included in that loop, to stabilize the transition state, because otherwise the reaction doesn't progress. If you have a lysine to alanine mutant, the enzyme is dead for all practical purposes. The rest of the residues form part of the core of the protein. I lost my cursor. Okay, it will appear again sometime. Okay, so we need lysine 201 to be in the correct position. Okay. Now, the issue is we only know how Cre looks when it's bound to DNA, not when it's alone. And that is a problem because then we do not know how the process starts, and we have no idea how we can regulate it and how we can make it less toxic, which has proven to be toxic. Now, work from Mark Foster's lab, this is an animal lab, has shown that the two domains are folded independently and they really don't talk too much to each other, and that is thanks to a very flexible linker over here. That is nice because then you can study each of the domains separately. But it's nasty because you cannot have a picture of this thing because it just keeps moving. In a recent work published from Foster's lab, what they did is to characterize the catalytic domain as a monomer, completely alone, without DNA. And it turns out that in that case, the N helix, that is the C-terminal helix, the one that invades the neighboring protomer, instead of folding back into its own nest, it folds back, but into the active site. So that leads to an autoinhibited enzyme until it comes close to DNA, and then DNA displaces the helix. Okay. They also did a deletion mutant where they cut it here, and while the domain remains folded, there are chemical shift displacements all over the place. These chemical shifts were just recently released to the public, so now we can actually do something about that. We can model them. Now, this gets more interesting. Remember that what we're trying to look at is this magical step of the isomerization. I was inactive and now I'm active, or vice versa. So the place where the N helix resides is right here, and the active site is right across it. So if you look at these two perspectives are rotated 180 degrees with respect to each other. This is halfway, 90 degrees, and you can see that they are back to back. So it's not very hard to imagine that what would happen on one side would affect what happens at the other. So the questions that we're trying to answer is whether this trans cavity is already formed in a monomer, if binding of the N helix is enough to organize the active site, or if you actually need quaternary interactions to accomplish that, and also if helix N forms different interactions in the active and the inactive forms. So our goal is to explore structural and dynamic roles of helix N in the catalytic domains, both in the active and the inactive formations. And as I said, we have plenty of structures to choose from. We got a synaptic complex where it is already assembled, but there's been no chemistry, so we don't have to worry about strange bonds. So the idea is to simulate the protein, well, the catalytic domain without helix N, with the helix in trans. That means that it's coming from another monomer. So it's really two molecules, it's a complex. And this one, we have to model it. This one is pending, I won't tell you anything about it. And these are the conditions for the simulation. We have 15 microseconds for each condition in 3 replicas of 5 microseconds each. So regarding the first question of whether the N binding site is present or not, looking at the simulations, looking at the RMSD with respect to the initial structure, we spotted a spike in RMSD and then it went back to normal. And when Marco plotted both structures, what we saw is this claw-like motion. It looks like a bear trap. So let me see if I can stop it. No, I can't. Okay. So there is this hinge motion, and this place is exactly the cavity for helix N. This is a microsecond range event, and it changes clearly the angles between helices of 2 subdomains, one that is over here and another one that is over here. Okay. Now, it could well be that we were looking at an artifact, because we know that force fields are not perfect. So we tried with an elastic network model with El Nemo, and it turns out that this particular mode, mode 2 and a little bit of mode 5, are enough to get the same type of motion. So it seems to be a characteristic of the module. Now, regarding the question of whether helix N, wait, I cannot see what that says. Crap. Okay. If it moves differently, whether it's in the active or the inactive conformation, yes, it moves more in the active conformation. Why would that happen? I have no idea. But there's an interesting hypothesis, it just occurred to me 5 minutes ago, that maybe these motions do something for the rest of the protein, and these motions are related to the catalytic process. This would be in line with Nusinov's idea of having a very rugged landscape, and you go choosing from one minimum to another, depending on what you have to do. Okay. Now, turns out that if now you just look at the distance between the distance and the angles between helices F and K on the one hand and I and L on the other, F and K are over here, I and L over here, without helix N, with helix N, what we see is that as expected, maybe, there are more conformations in the absence of helix, and that's okay. If you compare up with below, there are more hills up here than down here. But
Nina, excuse me, could you please try to summarize because you're running out of time?
I'll wrap up.
Sorry.
Okay, no, it's my fault. Okay, so again, active conformers sample more conformations than the other ones. If with the distances and the angles, we again confirm that it's mode 2 and mode 5. And regarding the question of whether we need quaternary interactions, yes, because this is the distance, a histogram of the distance between the lysine and the tyrosine. We need it to be on the order of 9 angstrom, which is around here. So you can see from these histograms that the population is negligible in the presence of somebody else. So this is a summary of what I just told you. I'm not going to read it to you. If you want it, you can have it later. The most important shortcoming is that we do not know if our starting structures are remaining where we put them or if they are migrating to something in between that is neither active nor inactive. So that's what we're going to check next. And finally, thank you for your patience. Money that allows us to buy local supercomputing. We have Mexican supercomputing, and then Carlos Amero is our NMR specialist, and Marco for doing all the work. Thanks.
Thank you very much, Nina. And our next speaker is Fernando Gonzalez-Nilo from Universidad Bello in Santiago de Chile. Fernando, are you ready?
Yes, absolutely.
Please, go ahead.
All right. Let me check how can I... Please tell me if you can see my presentation.
It's perfect. Now
Yes.
we are seeing not in the presentation mode, but in your... That's a good one.
All right. Thank you very much to Sergio and Guilherme for this great event. It's great to see how many works in this field. During the last month, well, during the last 2 years, we are beginning to work in antibodies again to SARS-CoV-2. The reason that because the pandemic has been pretty hard in all the world. So we are beginning to work on that. As you know, all of you know very well that the number of deaths is increasing day by day. Unfortunately, in the south of the hemisphere, the south hemisphere is going down right now. And we need to work in this important area. In that context, many teams around the world has working using basically this this kind of simulation, specifically the protein that interact with the target in human cells, in order to analyze what are the main what are the property and the energy interaction that drive this kind of recognition. In that context, many groups, including the show, for example, that is simulation done by this team, show that could be the spike protein. However, it's not so easy to analyze because, in fact, here in this simulation, they are using just a few sugars in the surface of the spike, but the truth is that the number of sugar around to the spike is a lot. That is the reason because the antibody doesn't recognize easily the nanobody, sorry, the virus. That is basically for us, the chemist, is basically a liposomes. It's very close to a liposome. So using this information and also a lot of effort done by different groups through the world, we are beginning to study the mechanism, how this protein interact with the ACE2, but also we was very interested in how we can blockade this interaction using therapy based on antibody. So therefore, in order to generate antibody again to the spike, has been not so easy, and unfortunately exists a number of information that is still not published. But anyway, exists some important information in the database of Protein Data Bank that with at least 3 different antibodies that interact with the RBD domain of this protein. And we are right now, we are working with this specific antibody through a collaboration with NIH. So that means not just make simulation. Unfortunately, we need a lot of work in order to purify the protein, as well purify the antibody, as well purify the RBD, that is the domain of this protein. Then when it's finally purified both proteins, we can measure the free energy interaction using this instrument, that's nano ITC. And also we can evaluate the enthalpy, entropy, CP, and a lot of information that is pretty useful for the people that is working with the molecular simulation. In this case, in this 3 case that is public right now, the key day is good. It's around to the nanomolar, the interaction that is good, but it's not enough. The KD should be in order to picomolar. In that case, we are just in nanomolar. Just a few difference in this KD is an important impact in the free energy interaction. So therefore, on the other hand, has to be highly specific. That's pretty complicated, and with a high affinity. That means near to picomolar. So to optimize the nanobody or antibody in general is pretty difficult. It's not easy because you need to modify the binding mode of interaction again to the target, the folding stability, that is pretty important also. The solubility, that is a huge problem because many teams has been related with the modification of a nanobody in order to improve the interaction, but unfortunately, the solubility decrease a lot and generate some aggregation. And then the density of the solution with antibody is so high that it's difficult to use as a therapy. The pharmacokinetic, of course, functional effect, etc. So in general, it's not very easy modify the banded interaction because it's a complex system, but at least the physical chemistry point of view, we could make some effort in that sort. Some bad news that the docking protein work great. That is look like a good news, but unfortunately just work well for dimers that already know. Yeah, all the program that we trying to use to evaluate the interaction between 2 proteins, unfortunately doesn't work well. Work excellent. Almost the 99% of the PDB can be re-ensembled because they are already crystallized. But if you put both a monomer and you make a small molecular simulation just to relax the lateral change, you lose a lot of the accuracy of the program. Therefore, it's a problem that is still not well solved. And also that is pretty important that the environment where the antibody work is very crowded. Yeah. Therefore, the affinity and the specificity is a great challenge. The good news that is increasing the number of antibody characterized with cryo-EM, but the bad news is that we don't have a cryo-EM in Chile at least. So I hope that in Brazil, maybe you can make the effort to have the first cryo-EM in Latin America. That I think that could be a great, give us a great opportunity to all the scientists that is working with protein engineering. In order to try to solve this problem right now, we evaluate using the classical method in order to modify the amino acid that interact in the antibody in order to improve the affinity. We are using the classical evaluation, the free energy interaction, using this method. It's not possible, unfortunately, to use a free energy or FEP calculation approach because they are really expensive. For this reason, we have to simplify a lot the method that we need to use to evaluate the energy interaction. And the same happens with the sampling of the lateral change. Even that is supposed that we can make some effort in using our current capability. I mean that using just a good GPU like a B100, for example, we can make to the year around to 1,000 calculations per year. So therefore, it's pretty difficult to cover all the probability of the interaction and all the variability of the amino acid that you have to change in order to improve the affinity. So for that, you have to consider the possibility that the amino acids that are part of the interaction contact region, you could change in order to improve the affinity, means through the improving the salt bridge in the case that is in the border of the contact zones, or improving the van der Waals interaction. That is the most important interaction when during this process of recognition, the molecular recognition of the antibody, again, to this part of the protein. So for that, just considering four amino acids to be changed, the possibility is quite high. So the number of permutation that we have to do is around to more than 100,000 different combinations that we could generate. Therefore, the evaluation, the energy interaction has to be improved a lot, but at the same time, you have to don't lose the precision of the calculation, because it's very the interaction is very sensible to this energy, to the evaluation, the experimental evaluation. So at the moment exist many approaches in order to improve the energy interaction, including the analysis of some loops, short loops using ab initio method, as well using the recent developed system by AlphaFold, for example, in order to improve the folding of the antibody. But still using this information remain a lot of work to do. Anyway, we expect them to make this matrix where we can evaluate all the combination, and then we can identify what is the best mutation that we have to do in the lab. All right. So in order to evaluate this interaction, that take around to 7 hours by system, but in very simple way. So the idea was explored for the 3 system, and the limitation has been recently mentioned by me, is that we can make just a few, 1,000 calculations per year in one GPU, but we need 100,000 of calculation. So just a few minutes rest on my presentation. Here is the implementation that we do in Amazon Web Services, when we have the possibility to make more calculation using a huge infrastructure, but it's expensive, unfortunately. And just to finish, finally, we make 8,000 of permutation in just 2.6 days. Will in our current capability, that the same mutation took around to 6.8 years. So again, the capability of the computation in this area is absolutely required. Even that, we analyze all these permutations in this tree system. It's around to 6,000 different variation in this tree system that has been evaluated right now. And in total was 3.6 terabytes of information and more than 40 microseconds. Now is the evaluation are ongoing in order to implement a easy system to test the presence or not of the virus. So some remark, the rational design is extremely difficult to predict, therefore is required an iterative theoretical experimental cycle. So it's necessary this integration of the system. Even the simplified energy interaction calculation require an important computational capability. Weak interaction, that is more important than I'm expecting, is still not well represented in many force fields. The recognition of the contact zone is still required modern effort, like it was done in AlphaFold, for example. The role of water in protein-protein interaction still is not well known, and it's difficult to implement in this kind of system. And I think that is required a more integration between bioinformatician tools with computational chemistry tools, in order to give us more opportunity to design new antibody. Thank you, Sergio.
Thank you very much, Daniel. So we have to really move to the next speaker, Laurent or Laurent. Are you there?
Yes. Okay. Laurent
perfect.
In French, and Laurent in Portuguese, Laurent in English.
Okay.
You can choose.
Sorry,
I will share my screen.
French is not my specialty. So please go ahead. I mean, the next speaker is Laurent Dardenne from the National Laboratory for Scientific Computing in Brazil. Whenever you want.
Okay, I have a problem here. Are you seeing my screen?
No.
No.
Just see a black image saying.
I have a problem here. I don't know why.
Then you have started screen sharing, but we don't see the sharing.
Okay.
Laurent, are you sharing a video or slides?
Slides.
Yeah.
There you are.
Okay. It's okay?
Yes, perfect. Please go ahead.
Okay. Good afternoon, everybody. It's I would like to thank Professor Guilherme Menegon for organizing this very nice satellite meeting. It's a pleasure to be here and have the opportunity to show you some recent works of the doctor related to the doctor project. In the last 18 years, we have been working on the doctor projects at LNCC in Petrópolis, Brazil. And the main objective is to develop a useful receptor ligand docking program for the scientific community. The first version of the program and the first version of the web server was released in 2013, and the second version of the web server for visual screening experiments was released in 2018. And today, the Dr. VS web server is a free facility provided by the Brazilian supercomputer Santos Dumont. Yeah. Doctor is a grid-based docking program where the energetics of the protein ligand is mapped in a grid covering the active site of the receptor. Originally, the program was developed to deal with highly flexible ligands. We developed a multiple solution genetic algorithm using a classical force field, the MMF94S, for pose prediction. And we also developed for binding affinity prediction empirical functions, different empirical functions using different machine learning techniques. All the receptor, ligand, cofactors, parameterizations are fully automatic inside the web server. Last year, we published a paper in JSAIMS where we investigated the capability of the program to predict the correct binding mode, the post-prediction for 53 protein peptides complex, where the peptides are very flexible. You have peptides with until up to 51 rotated bonds and up to 12 residues. And when we compare our results with the state-of-the-art docking programs described in the literature, we see that the results of the doctor program are very competitive, mainly with the GLIDE program, the SURFX program, and also AirDoc, that is a docking problem specially developed to deal with peptides. And in the right here, we have a system with a peptide with 31 rotated bonds, where only the doctor program was able to find the correct binding mode. We also developed our own scoring functions, and we developed many general for PPIs, for protease specific machine scoring functions, using different machine learning techniques, but we use only 6 physical based descriptors. Yeah? And we also evaluated these scoring functions in the test set, the version of the 2013 core set from the PDB bind. And when we compare our performance with the other scoring functions published in the literature, we find that also for the linear and the non-linear scoring functions, the scoring functions developed by our group are also very, very competitive. And in the last year, in the beginning of the last year, with the COVID-19 pandemic, my research group made a collaboration with Professor Marisa Fabiana Nicolas, also from LNCC, where to study more than 240,000 genomes from SARS-CoV-2. And the idea is to identify important genomic variants for the 6 main therapeutic targets associated with the virus. And all these structures, the wild type and the variants, are provided through the Dr. VS web server. Here we have, for example, for the main protease, we have two conformations, different conformations, and 2 variants for each conformation. We provided the structures. We have a total of 4 structures available in the portal. For the spike, you have 3 conformations, 1 complex with AC2 and one isolated from, but originally complex with AC2, and the other with antibody. And the same thing for the papain-like protein, the NS, the RDRP protein. We have also structures available for the nucleocapsid protein, and also for the endoribonuclease protein. Today we have our almost 60 structures available for the users through the Dr. VS portal. And here we have, for example, for the main protease, you choose the structures looking inside the active site, trying to identify important movements of the side chains of the loops, and to identify important structures. After that, we promote the mutations, and after that, you prepare the system looking for the correct proteination states and the possible flips of the side chain, for example, histidine or glutamines. This is a very interesting result where that reflects the importance of the choice of the original target. For example, in the left, we have the imatinib drug against the main one structure of the main protease. And for this virtual screen experiment, you have the imatinib ranked in the fifth as the fifth best ligand. But if you use another structure from the main protease, this same drug is ranked in at the 100th position. And in our experiments, virtual screen experiments, we always use both structures in an ensemble docking experiment. We also provide in the web server a data set of FDA approved drugs where we performed a proteination study, a macrocycle conformation study, a Autonoma study, and for each drug. And at final, we have more than the 2,300 molecules in your data set. And we perform a repurposing study using all the wild type for structures for all the 6 therapeutical targets, using the ensemble docking strategy, considering all the tautomers and proteinase states of the drugs. And we found some interesting results. For example, we found that elbasvir, for example, it's a drug for hepatitis C, is a multi-target for 5 of the 6 therapeutic targets of SARS-CoV-2. Ledisparvir, it's also a hepatitis C drug, has also a multi-target profile. And to use the doctor web server, you have 2 types of users. If you want to make a experiment up to 200 ligands, it's totally free. It's anonymous. You can use all the molecular targets provided by the web server. And but if you want to perform more than virtual screen up to 5,000 ligands, you have, and to have access to the FDA drugs data set, you have to submit a project for registration approval. And if you are approved, you can use the doctor for free. This the web server has been widely used by the scientific community, and in this year, until now, we have more than 24,000 jobs submitted and more than a half ligands evaluated by the web server. And here is the team, doctor team. It's a multidisciplinary team with people from computation, pharmacology, biology, physics, engineer, and it's out. Thank you very much. And we can talk at the end of the section.
Thank you very much, Laurent. Yeah, we will have, remember, we have a conversation after the session. But in this interest of time, we will move to the next speaker, who's going to be Marcelo Costabel from Universidad Nacional del Sur in Argentina. Are you ready, hey, Marcelo?
Yes, I'm ready. Thank you very much.
Thank you. Please go ahead.
Okay. Thanks, Sergio. Thanks a lot, Guilherme, for the organization of this nice meeting. Okay. Okay, it's not. Yes, no, okay. Okay, I chose this word for this talk because I think relates experimental and computational data, and also is an example of a multi-scale approach from quantum mechanics to coarse grain. And I think also that is a good story. The system under study is triatoma virus. The triatoma virus, TRV, was discovered in 1985 and is related with the Chagas-Massa disease because TRV is a viral pathogen of triatomines and was proposed as agent of biological control against them. In 1991, we wrote the first crystals of TRV, and several years later, we participate of the resolution of the crystal structure of TRV. TRV has an icosahedral structure with 15 two-fold, 10 three-fold, and 6 five-fold rotation axes, and precisely the five-fold axis of icosahedral viruses were suggested in a paper of 1987 as ion channels, like the ion channels in membranes, with atoms passing across, connecting outside and inside regions of viral particles. On the other hand, if we remember virus life cycle, we have several steps. One of them is when the virus penetrates the cell and releases the genetic material. And precisely, this mechanism is today unknown. So, our work aimed to determine the relation between the five-fold axis and the release of the genetic material from an icosahedral virus. Here we have a side view of the board with amino acid rings surrounding it. And so at this moment, we have the structure of the TRV and we have the hypothesis. And then we run a molecular dynamics to see what happened in the board. And when we run the simulation, we see that water molecules enter but not cross the region that we associate as hydrophobic gate of valence in this region. Sorry. Then, at this moment, we remember the crystallography. And in the crystallography, in the map of electronic density, we found a density that we associate with a magnesium ion in this place. And then we put an ion in this plate and run the simulation again. And now, in this simulation, we can see that the pore filled of water molecules, but never a molecule from the outside goes in or one from the inside goes out. Then we have water density maps about the wild type capsid, one mutation to verify that the gate is not steric but is hydrophobic, and the water density with the magnesium ion in the pore. Then, again, what about the pore? And at this moment, we remember another experimental result, whereas with atomic force microscopy, and when we increase the pH, the capsid and coating, and the genetic material is released. But for us, what does it mean to increase the pH? To increase the pH for us means that we have a lot of ions surrounding the virus. And then we can replace one water molecule for a hydroxyl ion. The next question that we have was whether a water molecule close to the ion can give up a proton to the hydroxyl and transform it into a new water molecule, and so on along the chain of water molecules. For verify that, we carry out the calculation of quantum mechanics for each step along the chain. Here we have not a simulation of, obviously, just a visualization model to show a hydroxyl going from the outside to the inside of the capsule in a growth slide mechanism energetically favorable. If you see, each water molecule give up the proton to the other closet, and the hydroxyl ion first at the outside, it's now inside the capsule. At this moment, we consider that if we can enter one ion to the inside the capsule, we can enter many ions. Then, in the last step to the multi-scale simulation, we run a coarse-grained simulation of the capsule TRB with a lot of ions inside. And we see that the volume of the capsule increased and the empty spaces are created between pentons, given the possibility of releasing the genetic material. Here we have a picture with the capsule with the volume increased and the region where the genetic material could be released. In conclusions, we found evidence that suggests a ring of valence belonging to BP3 could function as a hydrophobic gate. Second, we proposed that this hydrophobic barrier could be disrupted by a magnesium ion. We postulate for the pore a plausible role as hydroxide channel, and we suggest for the first time that structural features of the pore in the fivefold axis of TRB capsule could provide an insight into the plausible first steps of the uncoating mechanism of an icosahedral virus. Here is the reference of the article. Here is the people in the project. And thank you very much.
Thank you very much, Marcelo.
In the last, this is our university, and this is the people in my lab. No, yes, thank you.
Sorry, I interrupt you, Marcelo. Thank you very much for a nice and timely presentation. So we have to move to the next speaker, who's Mónica Pickholz from Universidad Nacional, from la Universidad de Buenos Aires. I think she will play a video. Yes, here we go.
Hello, everybody. Thank you very much for this possibility. It's a pleasure to be here. I will start with a slight change in the title, Interaction of ACE2 and spike proteins from different animals, before to get back to the original one. I don't think that we need to introduce these two proteins, are really well known, but just for the records, ACE2 is a membrane protein.
Sorry, Monica, but we have a blank screen.
Well, yes. In this work, we studied the interaction of ACE2 and spike proteins in a truncate model for 6 animals: cat, dog, pig, ferret, duck, and chicken, using
Monica,
molecular
can you hear
dynamic
us?
simulations. Here, we did 4 replicas of 800 each one. Here, I'm showing the truncated model.
No?
This is in blue, light
Sí.
blue, the ACE2, and in yellow, the residue binding domain of the
Monica,
spike.
your video is not playing anything.
This work is carried out by Sebastiana Dubiri, Eliana Sciuto, Galo Balatti, Carmen Domeni, Daniel Jacobson, myself, and Erika Teixeira Prates, alphabetical order. Actually, the original work came from Daniel and Erika Teixeira Prates.
Monica,
Well, we carry out these
there?
extensive simulations, and we come out with a lot of data that we are analyzing in the
Okay, I think I have the video here. Monica sent me a link, and I will play from here. Let's see
Okay.
if it works.
Hello, everybody. Thank you very much for this possibility. It's a pleasure to be here. I will start with a slight change in the title, Interaction of ACE2 and spike proteins from different animals, before to get back to the original one. I don't think that we need to introduce these 2 proteins, are really well known, but just for the records, ACE2 is a membrane protein. Well, in this work, we studied the interaction of ACE2 and spike proteins in a truncate model for 6 animals: cat, dog, pig, ferret, duck, and chicken, using molecular dynamic simulations. Here, we did 4 replicas of 800 each one. Here, I'm showing the truncated model. This is in blue, light blue, the ACE2, and in yellow, the residue binding domain of the spike. This work is carried out by Sebastiana Dubiri, Eliana Sciuto, Galo Balatti, Carmen Domeni, Daniel Jacobson, myself, and Erika Teixeira Prates, alphabetical order. Actually, the original work came from Daniel and Erika Teixeira Prates. Well, we carry out these extensive simulations, and we come out with a lot of data that we are analyzing in different tools and ideas. In particular, here I'm showing the contacts for the average for the 4 replicas of each animal. Going a little bit deeper, we look at specific interaction. We recognize, we find many interactions, but we recognize the patterns in mammals and birds. The lysine 417 interact with the GLU30, and the glycine 502 interact with the lysine 353. Take this lysine in mind, because we will get back to it later. And this is for cat, and an example for duck is the GLU36 with the tyrosine 505. Okay, we are looking to different similarities and differences between species and different properties. This is a work in progress, and the motivation was the zoonotic origin and transmission, and if the affinity of ACE2 are correlated with the infection of the animal infections. But we have another motivation, an extra one, that is exactly the title of the talk. Spike ACE2 interaction. What can we learn from animals? Doing simulation of the animals, can we find a pattern that help us to understand what happened in humans? Well, we know that the spike and the whole coronavirus suffer mutation along these 2 years. But what happened if we think in the other side, ACE2 as the one that is mutating, like checking the different animals and different affinities, we can learn how, if you have a game that coupled 2 different parts, and we broke one part in one, you will lose affinity. What happens if, how we can compare these structures? Okay, so with this in mind, I will enter an example that I think that will clarify a little bit the idea. This idea came from Erica and Daniel. They said that, okay, the spike, the amino acid 501 spike, that is an aspargine, was found that mutate to different amino acids in human, mink, and ferret. Why this happened? Or can we get some explanation comparing the animals with the human? They see that the amino acid in ACE2, the amino acid 154, was different from human, mink, and ferret. Let's concentrate just in human and ferret. Glycine, we don't expect that glycine do too much interaction. So we look who is interacting the aspargine. The aspargine was interacting with the lysine 353, its neighbors. So this interaction seems quite important. And this lysine 353 is conserved in the animals that we study and in at least in all the animals that I checked. So what is the role? Change the role in different animals. When we did the simulation with humans, we see here is the 501, and this is the lysine 353. We check to a specific distance, nitrogen oxygen, to nitrogen oxygen of 3 replicas, and we see that they were very stable in distance, around 4 nanometers, 4 Armstrong, sorry. But this changed completely when we look at ferret. This interaction is lost. They have different distances, and it was not any specific interaction. Seems that this interaction is less important in ferret than in human, even if the list 353 was not changed. What happened? Well, if we check the neighbor, the next amino acid of 353 in sequence is the arginine 354 for ferret and a glycine for human. This 354, this arginine, is also charged and compete to do a hydrogen bonds, for instance, with the lysine. So if we check in ferret, we see that the 354 in 2 simulations, 2 replicas, were doing hydrogen bond with the amino acid for 405. And in one, the 353 was interacting, but not with the 501, instead with the 495. So seems like this affinity with for the lysine 353 was lost for ferret, at least for this amino acid. This is a little part of the work that was already submitted for publication. It's under review, but you can find this in the archive. The authors were Michael Garvin, Erica Prates, Jonathan Romero, Ashley Cliff, Joan Machado, myself, Mirko Pavicic, and Daniel Jacobson. Well, I would like to thank you very much for your attention, and I would like to thank the financial agencies and the Swiss National Supercomputing Center for the computer time. Thank you very much.
Thank you very much, Monica. I would like to give the microphone to Guilherme right now, but it would be uncomfortable because he's sitting Thank you very much, Monica, again. And we move to the next speaker, who's Paulo Netz from Universidad Federal de Rio Grande do Sul. Paulo, are you ready?
Yes.
There you are.
Yes, I'm ready. Okay.
Okay. Whenever you want.
I have a video to share.
Sure.
I have to share the screen. And okay.
And are you using Windows or Linux?
Meantime, please remember that you can ask questions in the Q&A.
Share sound.
So we can address those in the end of the
This
conversation.
presentation with SARS-CoV-2 spike protein and relevant cell
Você precisa desligar o seu microfone. Dá um mute nele. Porque senão faz eco.
Turning public health today. These are the numbers of the pandemic in the last week.
Mas, Paulo, acho que você precisa ir no share
Oi?
pedir para ele no advanced e colocar para ele share a vídeo.
Sim, foi que eu coloquei.
É que o seu som está muito baixo. Acho que você abaixou então o volume do áudio.
Vou aumentar o volume do áudio aqui.
Isso. Mas muta o seu microfone depois, porque senão...
Só um pouquinho.
Sorry, everybody, for the Portuguese.
Have been invested in the understanding of the details of the interaction between the virus and the human cells. Huge efforts also been invested in bioinformatics-based drug repurposing and drug design studies, prioritizing natural products as phytochemicals. Considering the huge number of recent works, we apologize for not being able to give a comprehensive overview on the literature, but instead we will focus mainly on some recent contributions of our group concerning the study of the interaction between the viral spike protein and the human receptor angiotensin converting enzyme 2, ACE2. We will focus also on our studies on the interaction between natural products, specifically flavonoids and flavonols, with spike protein and other proteins relevant to viral infectivity. The coronavirus structure has been extensively described in the literature, and here we will mention some structural proteins: nucleocapsid N, membrane M, spike S, and envelope E proteins. In particular, the spike protein associated with cell recognition and fusion of the viral and cellular membranes has been the subject of several experimental and theoretical studies. A region of the spike protein, the receptor binding domain RBD, responsible for the binding with the receptor ACE2, was studied in depth by several groups using diverse methods. In the beginning of the pandemic, our group started to study the interactions between the RBDs and the ACE2, comparing the SARS-CoV and the new coronavirus SARS-CoV-2. We docked the spikes fragment using Autodoc, obtaining structures with essentially the same conformation as the experimental ones. The docking score of the SARS-CoV-2 conformation was found to be more negative, a stronger binding affinity. After that, we carried out 500 nanoseconds of molecular dynamic simulations for the isolated proteins and the complexes using Chromax with the amber force field. Based on the trajectories, we analyzed the stability, the dynamical properties, the network of hydrogen bonds, and estimating the binding free energy using several methods. Now the results. The hydrogen bond pattern of both coronaviruses RBDs are similar and can be roughly subdivided in three networks, localized in distinct regions in the interface between RBD and ACE2. However, There are more hydrogen bonds in the case of the new coronavirus, and they are mostly stronger. Using this analysis, some key residues of the spike proteins and the ACE2 receptor could be identified. The Gibbs free energy of binding with the receptor was found to be consistently much more negative for the SARS-CoV-2 for all the methods: docking score, sampled by ensemble docking, LI, and MMGBSA. Very recently, we applied docking and molecular dynamics simulations to investigate the effect of the mutations of some key spike residues on the interaction with the receptor. The mutations that we analyzed were the IN501Y, E484K, and triple mutation, also with K417N. We found that all these mutations yielded stronger interactions, particularly the mutation of the negative charge residue glutamic acid in the spike position 484 to a positive charged residue lysine yielded a substantial negative free energy contribution. Indeed, the E484K mutation and the triple mutated systems showed higher affinity, larger interface area, and induced stronger conformational changes. These are precisely the mutations found in some of the variants of the concern of the coronavirus. Now, we will discuss some contributions to the design of drugs intended to prevent infectivity. There are many targets for drug design considering the coronavirus proteins, namely the spike protein, the main protease, and the envelope protein. However, the entry pathway of the coronavirus can be more complex, and other human proteins could also play a role besides ACE2. In particular, the transmembrane protease serine-2, TMPRSS2, and the cathepsins CAT-B, CAT-L were also thought to mediate the entry of the coronavirus into the host cell and can be interesting targets for drug design. In collaboration with some universities in Turkey, we investigated, using several theoretical approaches, the interaction of flavonoids and flavonols with the proteins spike, TMPRSS2, cathepsin B, and cathepsin L. The protocol comprises the structural optimization of the ligands, download and immobilization of the receptors, and in the case of the TMPRSS2, homology modeling. The docking was then carried out with AutoDock, and the results were ranked, and the best docked structures for each ligand were analyzed regarding the pattern of interactions. The top-ranked ligands in the docking were chosen for further molecular dynamic simulations. These were the flavonoids epicatechin galate ECG, naringin NAR, peonidin PEO, and pelargonidin PEL, as well as the flavonols robinin, ROB, and gossypetin, GOS. With the ligand receptor complexes, we carried out molecular dynamic simulations with GROMACS using the amber phosphate. We analyzed the structural stability, RMSD, the ligand receptor distances, and the pattern of hydrogen bonds. Based on the molecular dynamics trajectories, the free energy of binding was calculated. To complement the study, the drug likeness properties and ADMET profile studies were also carried out. For most of the complexes involving the top-ranked poses from the docking, the molecular dynamic simulation results confirmed the strength of the interaction, with the exception of the PEO and PEL interaction with spike and TMPRSS2. Strong interactions were found for the flavonoids ECG and NAR, as well as for the flavonols ROB and GOS. A detailed visual analysis of the trajectory confirmed these findings. Regarding the spike protein, for which the interactions with the human receptor are well characterized, the ligands ECG, ROB, and GOS interacted directly or near the RBD, therefore could act as inhibitors. In the case of NAR, a migration away from the RBD was found, although the interaction remained very strong, with almost 4 H bonds. The interaction between the ligand ribonine and both cathepsins were also remarkably strong, with a large residence time and a large number of hydrogen bonds. The free energy of binding estimated with MMPBSA was consistently negative in the interaction with all receptors for the flavonoids ECG and NAR and the flavonoids ROB and GOS. As conclusions, the simulation showed a detailed picture of the pattern of interaction between spike and ACE2, explaining why the affinity of SARS-CoV-2 is stronger. Also, the spike E484K mutation yielded a stronger interaction, supporting experimental findings. Considering the interaction of the ECG, now promising drugs against COVID-19. ECG was found to be more effective than other compounds and also showed drug-likeness properties according to the Pincy rule of 5. It can be considered as a candidate molecule in the drug design against COVID-19, since it did not have any toxic effect on eukaryotic cells. Experimental studies are underway. Thank you very much.
Thank you very much, Paulo. And so we move to the next speaker, who is Roberto Lins from Osvaldo Cruz Foundation in Brazil. Are you okay, Roberto?
Yes, I am. Thank you, Sergio. I'm just going to share my screen.
Okay, yes, go ahead.
All right. So first of all, good evening, everyone. Thank you for the invite and thank you for the organized committee, which pretty much Guilherme in this case, or there was some help from Sergio and Teresa, as I understand, but it's mostly Guilherme. So yeah, it's glad to be here to see all your faces again, even if it's not I'm presential. We've been doing some studies on the on COVID-19 related type of stuff, because of course you're in the Osvaldo Cruz Foundation. I'm at the Department of Virology, and I'm going to try to show you some of these things that we've been doing. Our group is a protein engineering group, essentially, and I'm also part of the Fiocruz Surveillance Genomic Network. The talk is more or less in the same, it's similar to what another talk has been presented here. So the current status of the pandemic is that while we wait eradication, vaccination is finally catching up, but it's very regular throughout the entire world. So eradication is not going to to take place in the next few years. So we have to use vaccines. We're still doing some social isolation and some sanitary measures, which is very important, so we can at least avoid the surge of new variants of concern. Right. So in the meantime, well, we need several initiatives, since development of drugs or therapy, and even new, more cost-effective diagnostics that would be on par for quantitative real-time PCR. Thinking about that, we thought that we could develop some synthetic antibody-like molecules that could recognize the viral particles, right? So, and the spike protein seems to be a main target for both neutralizing antibodies, but also therapeutic innovations. So we thought that if we develop these molecules that can bind effectively to the spike protein, it could be, this could be used for acute phase diagnostics, right? Like PCR, but cheaper. In case of ELISA plates, for instance. And we directed these synthetic proteins to epitopes of neutralizing antibodies, well, we could potentially use these molecules also as therapeutic agents just as an alternative for monoclonal antibodies that are really complicated. So what we thought is that we could use a nanobodies as a scaffold, and in a nanobody, the hypervariable region called the VHH. So nanobodies, for those who don't know, these are the single chain antibody-like molecules that are produced by camelids and some shark fishes. So they have same binding affinities and neutralizing potential as regular antibodies, but and they are produced in addition to regular antibodies. So we thought that, but this hypervariable region, it's very stable, so it's convenient for us if we want to change amino acids. And we thought that we could use that as a framework and computationally designed new molecules that would bind the specific regions of the spike protein. And how did we do that? So the first thing that we did, we went to the PDB. We took over 60 VHH, in their native complexes, and we run a few hundred nanoseconds of molecular dynamic simulations. And then we characterized about 45 interfacial properties and looked how, these properties, the distribution of these properties. Then we did two things. The first, we fed these numbers and we have the experimental data for all this complex that we categorized as high affinity, medium affinity, and low affinity. And we looked then what are the high affinity properties, what they look like, their distributions for this VHH complex. This is one thing that we did. The other thing that we wanted to do, we just didn't want to design nanobodies that would bind, but we also would like to compare with experiments. So we took all these 45 interface properties and fed into a multiple linear regression algorithm, and we reweighted our free energy binding score. So you can see here that we have a correlation coefficient of around 86% and the standard error deviation of 1.6 kilocalories per mole. Okay, very well. So the next step was to try out the free energy binding function in a real world. So this has been shown here. So for the coronavirus, original coronavirus SARS-CoV-1, there was a antibody, this VHH72, that bound to the nanomolar concentrations, as you can see there in the left part of the screen, and it was neutralizing actually. So the nanomolar concentrations are already, it's enough to neutralize. But it did not neutralize. They tried on this SARS-CoV-2, and you can see that it's basically a 2 kilocalories difference. It's enough for not neutralizing anymore. Right? It's amazing. Then we did the same using our our scoring function, and we can see that the difference between predicted for the VHH72 for CoV1 and CoV2 is also 2 kilocalories per mole. So we're happy with that. Then we have chosen a couple of different epitopes, and we designed 1 million different nanobodies against these two different epitopes, so 500,000 for each. And not going to go into the details of the process, but it's all de novo design stuff. And we calculated the ones that we thought that would be most interesting to synthesize experimentally. So if you are right, the nanobodies would be able to bind the virus spike protein on their epitope of neutralizing antibodies, and would prevent the virus to then bind the human receptor. So first thing that we did, we calculate the binding affinity of these two antibodies against the receptor binding domain of the spike protein only. by microscale thermophoresis, and you can see at least one of them bound to this nanomolar concentration. This is it's 3 to the times to the 3 nanomolar concentration. So, but this is only the receptor binding domain. Then we also did 4 virus-like particles that were kindly provided by Sérgio de Paula from the Universidade Federal de Viçosa. He gave us this the particles, and you also measure the affinity, and you can see we have at least one of them that binds at nanomolar concentration. So we're kind of happy with that. And right now what we're doing, Fiocruz Pernambuco, it's also a reference for COVID diagnostics. So we have a lot of biological samples. So from the NB3, we take the inactivated nasal swab samples, and we are right now optimizing the ELISA plates to be used to detect the spike proteins or fragments or whole inactivated virus from these biological samples. The next step that we also do is to start with microneutralization assays, and but for that we require a biosafety level 3, and we have in our institute currently it's not operational, but should be operational in a couple of months. With that, I thank you your attention. I remain available for questions that some of our collaborators. This is the group highlighted people. Mateus Ferraz, bright PhD student who does all the machine learning type of stuff in our group. I also did the simulations in this specific case, and Camila Adam and Isabel Viana who did all the experimental work. And of course, I have to thank for the research agencies, and thank you for your attention.
Thank you very much, Roberto. And we are going to the next speaker of the session, who is myself. So it's going to be like half an hour or more. No, I know we are all tired, so I just changed the presentation. I did something completely general. Let me just open PowerPoint. Are you seeing the screen? Okay.
Yep.
So let me just take your time. Okay, fine. So thank you very much for again, Guilherme and everybody for staying there. I think when the audience has going down like half if we consider the beginning, but so instead of going too much into details of what we are doing, I think that at this point the best thing would be like to treat it as a conversation between old friends that don't see in a long time, which is essentially what we are. And so the 3 questions is where have you been, what have you done, and what are you up to? First question, well, it's a stupid question, having a home just as everybody else, so we'll go directly to the second one. What have you done? Well, essentially, I mean, all things considered, I think we were kind of fine. Just for those of you who don't know me, what we have done in the last like 10 years or so, I think 11 already, is to develop a force field for coarse grain simulations, which we call SIRA, stands for South American Initiative for Rapid and Accurate Hamiltonian. Essentially, it's a force field that contains moieties for simulating amino acids, nucleic acids. Actually, not all nucleic acids, it's only DNA. RNA will eventually come. We have parameterization for water and ions, and this year we also added metal ions because you can, I mean, nearly a third of the proteins that are reported in the PDB contain a metal ion. So if you really want to make a general force field, then you need also to have those as ligands. We have representations for lipids, and not so many lipids, but quite a few. I mean, you can have like about 30 or so varieties. So you can eventually also go and run simulations with proteins and membrane proteins, and eventually protein DNA complexes, and obviously protein complexes and so on. The important thing that started as a part of the strategy is that we use the same Hamiltonian that everybody uses in molecular dynamic simulations, this essentially makes makes it easy to run in any molecular dynamics engine. In particular, we ported this to GROMACS and Amber. It is quite easy to use. This is what people continue to say. And if you want to take a look, just go to here to this web page here, and you can download the package. And there are a few tutorials for how to do things and so on. So, closing the parentheses. What have we done in this period? Well, we made it to enter in the standard distribution of Amber. Special thanks to Adrian, who helped a lot with that. So now Syrah is also distributed in the standard distribution of Amber, and there is a spelling mistake there. So this is kind of one of the news. We have, of course, published a few papers. Just I don't want to into the details, but there is a recent one this year in which we assess the capacity of the force field to deal with IDPs, when it's intrinsic disordered proteins and peptides, and essentially it performs quite well, better than what we expected, because our force field, as many others, was initially considered to represent the folded proteins. So going to IDPs was something that was not granted at all, and we did a nice assessment. And eventually, if you want to go, you can read this paper by Florencia Klein early this year. It turns out that with the same standard parameterization that we published 3 years ago already, it works very nicely for peptide aggregation. We have another paper by Ezequiel Barrera also this year, in which we saw aggregation studies for polyglutamine or glutamine-rich peptides, but also for we took as a control all possible tetrapeptides, and we just saw that it correlates very well with results made in Amber force field at the all atomistic level. And also in a nice collaboration with Leandro Martinez, and I think it's a nice example of this initiative of a collaboration in South America and between South American people. We published a paper in JSIM in which we describe the kind of optimized way to pack viruses. The thing is that essentially you can combine some new options that appeared upon the collaboration between the two groups, mostly between Matias Machado and Leandro, to generate a new kind of packing options to avoid problems in the building up of the viruses. We applied this to Zika, and again, I'm not going to the details, but then you first go and pack the virus with Packball, and then you run the simulations with Syrah, and the results. I mean, for instance, the comparison against the data from cryo-EM, it's really quite good. What are we up to now? It's essentially computational biology, which is mainly what we are all doing. Not pretty much on the SARS-CoV-2, because I don't know, it's just kind of nothing we will like to stick there forever. But yes, we have published some things. One of those is an initiative that we took early in 2020 to simulate all the proteins that were published in the PDB, all the viral proteins. To my knowledge, was the first time that somebody ran simulations on the entire proteome of an organism and the very same conditions. All the simulations, trajectories, and everything were uploaded to Zenodo, and the whole thing was published in a special issue that Fernando kindly invited us to contribute to a special issue in Frontiers. So you can go there, and eventually, if you're interested in coarse grain simulations, there are 10 microseconds or more of virtually any protein. And if you want another protein, in another viral protein, of course, we can just run the simulations for you. Just drop us an email. There are some stuff going on with the spike, and we have published a paper recently about the dynamics of the spike protein in collaboration with some colleagues in Chile, in which we just picked up something from the dynamics and also from some mutations arising in the southern region in Chile, in Magallanes. And this, I won't go into details, but I'm really tired about this virus. So finally, what we are now moving towards is really computational virology, which is not just running simulations on viral proteins, but really try to understand the dynamics of entire viral particles. This is something that we can do. I think is at the best compromise between cost and resolutions with our force field using multi-scale simulations. We have currently at least 2 projects, one running with flaviviruses, and this is another thing I would like to mention. It's something we did thanks to a grant, a shared grant, in which we could use computers in Santos Dumont. And we have another very recent project in PCV. It's a porcine virus, in which we made it to predict some mutations that overstabilize the capsids. And this is, before ending the presentation, I just eventually would like to tell you that we'll have some open positions, hopefully for the next year, once the traveling restrictions ease. So eventually we will be sending you some calls. We want you for Syrah force field. And with this, I will just like to thank you. I think we are again all tired, but I will really like to thank Guilherme again to took this burden to organize the, I don't know if this is the 4th or 5th SAIMS meeting. 4th, thanks, Guilherme. To the attendees who stayed until the end, and to the guys who did the hard work all these years. And while I was preparing this rapid presentation, I was thinking whether I have to put or not some founding agencies. But actually, the project was I never made it to get the grant for that. Only the salary is paid by the institute and computers and computer time. Perhaps I should have put Santos Dumont here too. But with that, I would like to stop the presentation. Thank you very much again, and look forward for a final discussion, and see you as soon as we can to share a glass of wine or caipirinha or whatever. Also, pisco sour will be fine. Thank you very much.
Great, thanks a lot, Sergio. So I guess we have come to an end for this second session, and we are open for discussions. I think I'll try to open everyone's call to unmute. So I hope everyone is able to speak now if they open their microphones.
Okay, I have a question for Sergio. Amongst your list of favorite viruses, have you considered adenovirus?
Yeah, no, honestly, no. The reason why we were, I mean, well, coronavirus, of course, the reason is obvious. The reason why we started running simulations with Flavis was because of the epidemics here in South America, mostly in Brazil, with Zika. And the PCVs is because it's very easy to run the simulations. They're the smallest viruses, and they have a huge economic impact in practically worldwide. Like it's another pandemic, nobody knows about it, but like between 10 and 15% of all the pigs in the world are infected with this virus. So we are trying to run some. I mean, and we kind of made it to predict some mutations that will stabilize the capsid and eventually will be of use as a vaccine. But no, on adenovirus, no. We could consider that. But
it's a vector for vaccines and stuff like that, and it is nicely complicated inside. So you would love it. Thanks.
I have a question, a couple of questions myself.
Yeah.
To Laurent. Laurent, all this docking, the dockings that you or people around the world have been doing on Doctor are available on the website. I mean, I'm thinking in people just running, I mean, perhaps going and fishing for stuff that is already done, like with compounds that have some small modifications, or where you can look for chemical similarity.
No, because you have to guarantee the secret of the submission.
Right.
It's not possible to perform this, and we have also some researcher from AstraZeneca and other companies asking us if you have a software for local running, because they didn't can run their molecules from a company inside a web server. But until now, we don't have a software for a local computer, because there are a lot of problems. Because if I give a software, a code for local running, probably in the next week, I will have a lot of emails in my saying your software is not running. I don't arrive to put your software to run in my computer, and I'm not a company to perform this type of work. And for us, in the moment, we prefer to only make available the doctor program using the computer facilities of the Santos Dumont supercomputer.
Thanks.
It's me. Laura, I was curious about some points from your work. I'm not sure if I lost some details in your presentation, but cyclooxygenases are very high glycosylated proteins embedded in membrane. So have you considered these effects? Because they could interfere in conformations. I think the peroxidase site increases a lot in surface with the glycosylation. And another, a second comment, I'm not sure I missed this point, but the heme group is covalently bound to a histidine, to an axial histidine. So how the arachidonic acid can displace it from the catalytic site?
Okay. First of all, during the simulations, we maintain a restriction in the distance between the ferric and the histidine. So there was no possibility in that simulations of having a displacement of the heme group. So what we did was checking with electronic structure calculations the strength of the interaction. But we should do a simulation, a QMM simulation for that, or something different. So it was something like seeing if you introduce your arachidonic acid in the non-productive disposition, it seems that the interaction between the histidine and the ferric is going to be debilitated. Okay, that is for the part of the so in the simulation you cannot see the heme group going out of the protein. So we have a initial evidence in that direction. The thing is that there is a hypothesis from the empirical part that sustains that you have a hemicide. Sorry, because I'm very tired because we have a crisis in our faculty in this moment. So I'm following with the two things in parallel. So it's difficult for me now in English, but I'm going to try. The idea is that you
Bien
have...
también en español, sin problemas.
¿Ah?
Está bien también.
¿Puedo?
Exactamente.
Puedo. Agradezco en este momento. Me facilita. El tema es el siguiente. Lo que nosotros tenemos son indicios iniciales de debilitamiento de esa interacción. Lo que tendríamos que hacer sería una simulación QMM que permita la rotura de esa interacción, o una optimización de geometría para ver si en la optimización cómo queda el enlace y después seguir por allí, por ese camino. Eso estamos empezando a hacerlo, no lo hemos hecho. Todo lo que yo mostré se basaba en simulaciones clásicas y que partían que donde estaba la presencia del grupo hemo estaba con una restricción que no permitía la salida, porque si no se salía inmediatamente y no veíamos toda la reestructuración. Con respecto a la glicosilación, sí, y no solamente el tema de considerarla. Lo que se sabe de los experimentos es que en algunos de los residuos es variable la glicosilación, y eso sí incide mucho sobre la estructura. Nosotros tomamos de la estructura de ratón, la única que hay, que viene determinada con una serie de moléculas en la cristalografía, la limpiamos de eso, y donde había glicosilación la dejamos. ¿Ah? Y es la estructura que está disponible, creo, hasta donde manejo, no he visto que saliera ninguna más. Hay 80 y tantas estructuras de estas enzimas, pero de la de ratón es esa la que hay disponible con el dímero. Después hay monómeros y otras cosas más que también estuvimos viendo. No sé si esto contesta las dos preguntas que planteabas.
Pero es sencillo también construir la glicosilación cuando se sabe la posición de inserción. Como se sabe la estructura de glicosilación, es fácil también complementar y hacer toda la árbol, pero certamente, gracias.
De nada.
I promoted Horacio Guzman, originally from Bolivia. I think now are you still in Germany, Horacio? Who had a comment.
Hi, Sergio. Yes, I'm still in Germany, so it's very late here.
Please go ahead.
It's quite late. I had some questions for Pablo Dans, but I think he's not in the panel, right?
No, I think he left.
Okay, so I will try to email him directly.
I have a question when Horacio is done, but I cannot raise the hand. I don't see the button here, so say.
Okay, you're allowed.
I think as a host, I don't have this.
I am done. Go.
Can I? Yeah.
Yeah.
It's a question to Nina. Is she around as well?
She was.
Yes, I'm around.
Okay, Nina. I wrote in the chat, but I didn't see any reply, so I'm saying it again here.
Sorry, I missed it.
Yeah, no problem. So in your phosphate transfer reaction, you see the transition state stabilization, and you mentioned that there was a lysine there that was essential to stabilize the TS, but there was also an arginine there. Why is not the arginine enough? I mean, there are loads of phosphatase enzymes that are stabilized at the transition states. It's well stabilized by only an arginine. There's no need for lysine.
The thing is that this lysine talks to the 5' OH that becomes the leaving group for the reaction. So what you want this lysine to do is to work as a proton donor.
Okay.
So that's something that an arginine can also do, but it's going to be more painful. The pKa for an arginine is around 12 or something like that, and for the lysine it's closer to 10.
So it's not electrostatic, it's more acidic catalysis. Okay.
Yes, right. it works as a general acid-base catalysis.
All right, thank you.
Bye, people. I have to leave.
Mauricio, do you have a question? Mauricio,
he's muted.
Yeah.
You're muted.
It's okay. I have one question for Sergio, which is, I actually have two questions. First one is that given that you use the same force field, mass expressions for atomistic one, can you mix a fine grain and coarse grain for bound systems, or which is the hardest case, or for non-bound systems like a protein in a membrane? Can you do that?
Yes, I mean, there are two kind of dual or actually triple scale representation. One is for a solvent, so you can have an atomistic solvent with a thin layer of water, and then coarse grain solvent, and then even supra coarse grain solvent if you want. This is what we use for the viruses because it works well when you have a lot of solvent, and if you have a virus capsid, then you have a huge amount of empty space in the middle because, to my knowledge, there are no structures of viruses with the nucleic acid salt because it's difficult for the symmetry treatments and everything. So this is what we use, and this is why it's so economic to simulate these kind of things. And the other thing that works at 2 resolution is DNA. You can have a half, say, of DNA at the coarse grain level and half of DNA within the same double helix. You can have pieces at the coarse grain and pieces at the all atomistic level. Yes, this is implemented and it's very easy. I mean, it's straightforward to run. For proteins, not yet. I mean, we can't have yet half a protein coarse grain and half a protein all atomistic. There's somebody who has. I mean, there are a few things that a few people who have done that. We are not yet at that point. What about it's not impossible, but
what about the membrane coarse and protein fine?
No. For lipids, it's only coarse grain. We haven't checked. I mean, we did, but very long time ago, before we have a good representation for lipids. And the problem is that you have start to have some effects with interfacial pressures. So essentially, what you have is that the bigger phase pushes and generates a kind of a droplet, or either of lipids or water or whatever. So eventually, what you have is an effect of pressure on your solids. So it only relaxes if your system is huge, but if your system is small, because it's like it goes with the 1/r of where r is the curvature of the meniscus, then you really have to go to huge systems to not have problems. But yes, the interfacial pressure is an issue. It is an issue. I mean, it is a physical thing. It's not related to the force field. Whether you have an interface, you have an interface pressure. But yes, in principle, I mean, we haven't done for proteins and membrane. I think the group of Essex have done this with ELBA. The name of the force field is ELBA, electrostatic based something.
Yeah, I don't see any hands raised, but before we finish. Oh, okay, sorry, Sergio. Yeah,
yes, I have a question for Roberto. Roberto, at a certain point in your presentation, you said that you generated a huge number of proteins. You mean in silico?
Yeah, in silico.
And perhaps a naive question: it is not easier to get a llama and let them to do their hard job and then to purify the good ones?
No, it's well, see, you have to have the animal lab, and you have to have the llama, and well, it's and at the end, when you do experimentally, you don't know why it works. There is also this type of thing. So, and in silico, within 2 weeks or so, as I said, we can generate, like I said, a million structures and triage them. So in less than a month, we have, a few candidates. And so I think it's actually faster to do in silico than using a llama.
Okay.
And because if you do the llama, you also have to triage afterwards in a lab, and you have to triage a lot of them. So it's actually very expensive to do. And besides having the appropriate animal farm or whatever, that with all the appropriate safety levels and sanitary levels, I don't think it's easier.
Okay, thanks.
Welcome.
I have seen Munir. Munir, you can.
Yeah. All right. No, I just have a question for Sergio. I wrote in the chat because, well, I wanted to know, Sergio, if it is. I know you have a parameterized force field for the proteins, of course, for the amino acids and for the metals, for some metals. But I wonder if how hard would it be, for instance, to parameterize other, for instance, phosphate groups or hydroxyapatite, for instance, which are important for some biological functions, especially modeling of bone-like tissues.
Yes, I mean, the thing I did, obviously, because of time reason, didn't mention is that the way in which we parameterize things essentially is based on statistics from the PDB. So if you have enough statistics, and of course for proteins you have a lot, but for small molecules, and it may be tricky. So we don't have a kind of a recipe like, I don't know, Martini. They say, okay, this is 4 atoms to 1 bit and go, which doesn't actually work that well. But we don't have a recipe. Having said that, if you're interested in kind of functional groups, chemical functional groups that are already present in the force field, then that would be easier, much easier, because essentially we have proven that they work like Legos, and you can interchange them. So for phosphate, of course, we have a phosphate in the DNA, and it's the same parameterization that we use for the phospholipids, for instance, just to say some. So in principle, what we would need to do the parameterization is some statistical information about the interactions you want to reproduce.
Marvelous, thank you.
Sergio, let me just clarify one more thing that I didn't mention, that there is another round of things that we do, because as we intended to, that it could be used as a therapeutic agent. So once we do the design for the binding, we also redo the design of the whole surface in a way that we call it, we try to humanize it, meaning that using we put back side chains that are very similar to the ones found in human antibodies, so it doesn't give immunogenicity if you use them in humans. So that I also forgot to mention.
Yeah, thanks.
Mauricio, I think you're trying to.
Yeah, I have a question for Fernando Gonzalez-Nilo. It's more of a practical question that you said that you run the simulations on the Amazon Web Services, and that has basically infinite CPUs. But there's a lot of discussion in Brazil what kind of model HPC should follow, and I was curious about to know it. I don't know if it's a sensitive point, but is it very expensive, and is it competitive to have a local and local infrastructure? Could you comment on that? Why you decided to run on Amazon Web Services? You don't need to be very specific, but just.
No, I think that is a great question. In fact, I have writing some question about that, the same. So the first, I think that in Brazil, I think you have a great opportunity with Amazon, because the earthquake of Amazon is there, and to us, give us a grant. So in fact, to University of Chile, give them to $1 million of grants. In our case, was just a few $1,000s. And then right now we are implementing Amazon Web Services I think that is competitive in the context, in the total cost. I mean, because we have our own infrastructure. We are in a private university, and the cost of the power, we need to pay it. We have to pay everything. We don't have a support from the state, that in the case of any university, a public university, that maybe is your case. So in that context, I think it's worked well. And I think that, and also you have the possibility to use a huge infrastructure that they have. I mean, they have the last GPU. Now they have the 800 that is great for molecular dynamics. In that work, we use just the B100 that also is working very well. But you have many possibilities to organize your work in several GPUs at the same time. So in that context, you can compensate the cost that you have to pay the company versus to have your own infrastructure. Also, well, everything related with the power of the building is complicated. Sometimes you have some power problem, no? And then you have to reinstall everything and put the calculation again. In that case, it doesn't happen. In general, it's quite equilibrated. It's quite equilibrated. Is my opinion.
Roberto?
Desculpa, esqueci de baixar a mão.
Okay, so before
Sorry.
we finish, and I think that I have a few break in the questions. I really would like to say a couple of things. First of all, I would like to thank all of you, all the participants, all the speakers for the second session as well, for being here till this late time. It's almost like we've been here for 6 hours already. So it has been a long afternoon and night here in Brazil already. I also would like to say that some of you said thanks for the invitation when you're starting your talks, but none of you were invited. This was a hands up. Those who rose their hands first got in. So I haven't invited any of you. So I would like to mention that because all the people watching this might be thinking, why Guilherme has not invited me to this? So if you're feeling like that, that's because you are not in the SAIMS email list. So please subscribe to that list and participate in a email exchange there. And next time you will be here. Okay. So again, thanks a lot to everyone. And I apologize for those that could not give a talk, like Munir, for instance, that took a little bit to raise his hands in the beginning of this, the process we made. There were a few of you who got late as well, so could not find a slot to speak. I'm sorry for you, but we had 24 speakers today, so it was really the most we could do, 10 minutes each. So but anyway, I think it was a great meeting, and I thank you all for the participation, and thanks a lot.
Thank you, Guilherme, for all.
Thank you, Guilherme.
Thank you.
Thank you.
Nice to see you.
Thank you. It was so
you.
great to see you.
Yeah.
Who knows, we can get together next year in person.
Yeah, in person. Yeah.
Yeah, absolutely.
Bye-bye. I hope so.
Okay.
Everyone.
See you guys later.
Very nice.
Somewhere near Ecuador, so that it's South America, including the not so south.
Yeah.
We tolerate you,
Yes,
Nina.
of course. Latin America.
A comment, just a short comment. Unfortunately, it just has been observed a new virus. So unfortunately, in a few years, we will have a new pandemic scenario. So that is very important that maybe our community has to make a great effort to be more prepared for the next generation. The virus has been already detected in Hong Kong, and it's and unfortunately will be more dramatic because it's more effective in children. So I think that as a scientific community, we have to make a great effort to be prepared for the next time. Sorry.
Do you know what type of virus is it? Do you know, Fernando, what type of virus?
Yeah, it's a coronavirus also. It's that has been well contained in Iceland. I know very well a colleague that is working on that in NIH, and he told me that just in the last 20 years appeared the 5 more aggressive virus just in the last 20 years. So 5 great virus, and the last one, the current SARS-CoV-2, was the pandemic event. But he thinks that is coming another pandemic that would be more dramatic because the vaccine that in fact many